Good afternoon, sir.
Good afternoon. Manaswini, Namrita asked all to join.
Good afternoon, sir.
Good afternoon, Vivek.
Yes, I'll ask.
Okay, so good afternoon all. So now we can start with what we have done yet. And as we were talking about the module 1, and that constitutes your enzymes. And in that we talked about the bit history and the classification of the enzyme that you use. And these enzymes, you also call them the molecular tools. Among them, the first we are talking about the nucleases, and then we classify what nuclease is, what they do, and how they do, and what their type is. So the nuclease, we mentioned that there are two types: one exo and second endo. Exo and endo on the basis of where they are cleaving. Then exo, how they are cleaving, both the strand or only the one strand, single strand or double strand. On that basis, we describe them. Then we talk about the endonuclease. So endonuclease means within. Within simply means two nucleotide inside. Then in the endonuclease, the most important one was the restriction endonucleases, because the restriction means describe the specificity. Specificity means where the location where they're going to produce the cut. And when we're using term cut, this term cut simply means the breaking of phosphodiester bond. And we also mentioned that there are two types of the property. One is the modification and second is the digestion. And how they're being discovered, that is the protection immune system of your bacteria. Then their property, one property is the recognition site, what they are recognizing, that is on the DNA. And what are the property of recognition sequence? There may be 4 to 8 bases long. Palindromic natures they are. Most of are the palindromic, but it's not necessary that every time it is going to palindrome. But most of time it is going to palindrome. And cutting type, symmetric and asymmetric is also there. That is the property of your endonuclease. Then come to the type of restriction endonuclease on the basis of their property, their nature, their recognition site, their composition, their requirement. We classify them into 1, 2, 3, and 4. And then we talk about the 1, which is cutting in the remote location. And 3 also cut at the remote location, but the remoteness is going to be changed. Remoteness in case of 3 is less, whereas in case of the 1 is very large. Then come to the most important one, that is the type 2. And type 2 one is cutting within or just very near to the your recognition site. Their requirement is just magnesium chloride, no other requirement it is having, and it is having only one function, that is a restriction. There is a breaking the function, restriction digestion. No other function it have. Then we talk about type 4. So type 4 is another type, and type 4 is one which can digest your modified DNA sequences like methylation. So methylated sequences are protected from the restrict enzyme that we are talking about. But here in this case, if they are methylated also, so type 4 can do produce cut in those sequences also. Then come to the type 5. So type 5 is the new system, that is we call them, that is a CRISPR-Cas9 system. That is also a type of endonuclease, so their mechanism is a bit different. their recognition is not done by the enzyme itself. In that case, the recognition is done by an RNA. So RNA, we know that it is a single-stranded sequence, so complementarity is there to go and pair with the sequence which over the DNA, which is complementary to it, so that you recognize it, then it produces the cut. Then the sixth type is going to be your artificial. Artificial means we design as of our choice. We design if we want that this sequence I want to recognize and this location I want to recognize. On that basis, you can design it. You can engineer them. So genetic engineering that are FRDT, the recombinant DNA technology based enzyme that you come under the class of 256 that we call the artificial enzymes. Then we discussed what are the basic differences. So basically, as you mentioned, four basic differences are there: their composition, their location, their requirement, and their function. How they do, what the function, first they bind to DNA non-specifically, like DNA binding domain is going to be there, is on the enzyme which will recognize the DNA. Then to the specific one, the sequence which is recognized, and then the third one is the catalysis, that is the breaking of the phosphoester bond. Then we talk about what type of cut they produce, and we mentioned the blunt cut means for both the strand they are cutting in the same location, that leads to the production of a blunt end. And second is a staggered cut. In the both the ends they are producing cut at a different location. That is called that produces sticky ends. And sticky means they are leaving the overhang, and the overhangs, or simply you can say the single-stranded hanging regions. And these single-stranded hanging regions are those having the property of forming hydrogen bonds. That's why with that they can stick to that one. Then different types classification of enzymes came. So different enzymes are there, different recognition sequences are there. So these are the must. You must remember some of the important enzymes. There are some important enzymes, but the recognition sequences, at least 4 or 5 of these you must remember them and their source and their recognition sequence. And then come to the naming, how we name them. So naming is Smith and Nathan, they describe its naming, and name have their source and their identification number also, source, string, and the number, 3-4 component it have. So source simply means if it is a E. coli, you can say, and then the Shishakula. So E is going to be there for the genus, O is there for the your species, and this eco is being derived from your specific name. So that's why it is being italics. So here also in the name of enzyme, these first 3 letters they must be italics because they are derived from the species. And the 4th digit is going to be your strain. So the R strain is there. So different strains are there. So on that basis we name it. So R is the strain of E. coli. And this is the 1. This is the number of enzyme equal 1 in the first enzyme, equal 2, equal 3, equal 4, 5, 6, so on the numbers going on. Here you can see the hint 3 is going to be there. HI in hint is the your genus Haemophilus influenzae, where D is the strain, RD, so D is the strain name, and 3 is the third gene which being isolated from it. Similarly, in F1 is there, so the genus and species same for in F, wherever just a moment. Okay, so as you are seeing here in this case of Hen F, Hen is the same species and genus are same for these two, and wherever strain is going to be changed, so F is the strain in this case, so that's why you are having 4th one is the F, and first is the gene, so there is a Hen F1 is the name, and Hen is going to be italicized because it is derived from the genus and species of your source organism. So this is about the nomenclature. Then we talk about star activity, that is recognizing a non-specific sequence, a non-specificity, that is there in your enzyme that we call the star activity. Then we talk about different isomeric forms. Isomerism here is on the basis of the recognition sequence or the type of cut they produce or the source you are going to get them. So for that you are having the isosisomers, neosisomers and the isopodamers. So they are the different types we can discuss. So then we talk about the application. So in this lecture we were at this point, and from now onward we have to move further. So before moving further, I would like to ask you, any one of you have any doubt or any clarification you need, please let me know so that we can move further. So is there any query regarding till now what we have done?
No, sir.
Okay, so that's great. So if you don't have any query. Okay, so then we are talking about the application. So this is the main thing why we are talking about these enzymes. Why these enzymes are so important that we spend at least I think 2-3 days we spent on this enzyme. So this is a genetic real engineer that we are talking about of your system. It is going to be your restriction engine. But the job of engineer is the redesigning. So for the redesigning, first thing is you should cut it into the proper way. So simply if you know that a carpenter is there, so first thing is a carpenter is to produce your wood in a proper shape. So when it put into the proper shape, then the job of nails are going to be there to join together. So the first in your system is going to be these restriction enzymes. They are going to produce segment that you need of simply your DNA of your choice, segment of your interest or sequence of your interest that you need, and so with that you can use it. So simply it have various applications in your genetic engineering. Simply as I mentioned, it is cleaving. So wherever you have the cleaving role, you need restriction enzymes. They are used to process insertion of genes. So if you want to insert a gene, so you have to process it. So for the insertion, you have to digest your vector. You have to digest your gene of interest. So this is you need restriction enzyme in that case. They can also be used to distinguish the different alleles, different polymorphisms. So as someone of you asked, what is the SNP, single nucleotide polymorphism? Here that you know the name. In detail, we will talk about it in next module, I think. But as the name is there, single means one, nucleotide means your ATGC, and polymorphism is more than one. Means simply at a one location, one nucleotide may be at that location, maybe A, maybe T, maybe C, or maybe G. So the variation at a single variation of single nucleotide at a location is called SNP. So simply, if you just before going to detail, just want to say, like if you have any gene inside you, so your gene, my gene, and any of other genes, if our same gene, like a gene of speech, like our everyone is speaking differently, everyone's speech is be different. So why everyone's speech is different? So that may be because of some nucleotide variations are there that make our gene to express differently, to function differently. Same gene function differently in different individual. So that is some SNP, you may say that. So if you want to identify SNP, so this polymorphism is going to bear within this restriction site. As you know that like if you talk about EcoR1, so EcoR recognition site is GAATTC. So if in case of T, if C is there, so in one individual it is the T, in another individual it is the C. Then what is going to happen? In the individual, it is having T, equilibrium is going to produce a cut, and in individual, there is a no T, there is a C, then this equilibrium is not going to produce a cut. So simply by this way, you can say that that gene is in a different form. Okay, so this is the one application of it, and one more application is your restriction fragment length polymorphism. So simply as I mentioned previously, SNP. So if the polymorphism is there, so that polymorphism leads to one more level of polymorphism that is called the fragment length polymorphism. So So as if you remember when we were talking about the probability of finding restriction site. So if the restriction site is of 4 nucleotide long, then we said that the 4 is to power 4, so simply 256. So every 256 nucleotide there is going to be one restriction sequence. So this is a probability. So it is not always true. So in one individual, maybe to at every 256 one sequence is there. In another, maybe that after every 256 not the one sequence. There is a at 800 you may say that. Okay. So two individuals like you may say that one of your student anyone simply is called the Adish. So in Adish, if you talk about a gene, the gene that is a salivary amylase gene, if you are talking about. So salivary amylase gene is a gene that have in your one, we are having a one restriction site at 256 nucleotide sequence, but in my case, it may be presented at 800 nucleotide sequence. So this is something you may say it's a polymorphic pattern. When such thing is going to occur, in that case, the fragment that you are going to get after the action of your restriction enzyme, the size is going to be a bit different. So in detail, what this RFLP is there, I think we are going to talk about this in detail in your module number 4. And this is the technique, this is the RFLP is the one which we are going to utilize when we want to know the paternity disputes. So whether the kid belongs to the father or kids belong to the mother or not. So whether the father is a biological father or a genetic father or a true father, whatever your different terms are there, we'll talk about this later. Okay, so this is a bit few application of restriction enzymes, and even simply if you talk about to say diagnosis, so you can also use restriction enzyme in the diagnosis. Okay, so this is about the first enzyme, that is the restriction endonucleases, and what they are, what their role is, and what their type is. So now we are moving to the next enzyme. That the name of that enzyme is the ligase. So as I mentioned, when I took the example of the carpenter, so in that case, the carpenter as mentioned, the first thing is you have made all the wooden components, wooden parts. Like if you want to make a chair, you made the you cut down your legs, then you cut down your back and the seat of your chair. But now you have to fix all these together. So fixing, you need nails or you need adhesive, simply the Fevicol. So here also in the restriction enzyme, what they are producing the fragments. But when the fragments are of no use, when you want to engineer the things, engineer means not just the breaking. Engineer means the rearranging and rejoining in a way that you can use in a fruitful manner. So here also the role of the ligase is there. So DNA ligase, what they do, their job is the joining. So joining, the role of your restriction enzyme was breaking the phosphodiester bond, but here is the reverse. Here the job is making the phosphodiester bond, formation of the phosphodiester bond between two deoxynucleotides. So as we mentioned that in previous case, so when we break a phosphodiester bond, after the breakage of phosphodiester bond, what you are going to get? You are going to get a 5' phosphate group and 3' OH group. So here what you are going to do? Here you are going to make it the phosphodiester bond. So for the phosphodiester bond, what you need? You need a 5' phosphate and 3' OH. So these are the requisite of your ligase. So ligase, what they do? They catalyze the formation of phosphodiester bond between two hydroxynucleotides. So what they require? They require a free hydroxyl group at 3' OH. So simply we are calling it the 3' OH. So here I want to ask you one thing. You are all of you have done your molecular biology course, and I think what is this? Why you are using term prime? Why we simply not call the 3 and 5 and why we are using term prime? What this prime mean? Why we are using prime over this 3? Can anybody tell me why we are using this term prime? Anyone? Manishwini?
So I don't know the answer.
Vivek?
Sir, so is it regarding the position of the carbon in the structure?
So that you can tell by just by 3 also.
Yes, sir.
Then why we are using term prime?
Sir, is it regarding primer?
No, it is just your whether your sequence D primer is there or not.
So
You know
is it
DNA
because of
stands
the
to
activity of
recognize.
DNA polymerase?
Sorry, no,
Is it because of the activity of DNA polymerase?
no, but you can prove that a 5 to 3.
Sorry, sir, I didn't hear you.
You simply can call the 5 end and third at 3 end. You are using term prime there, 5 prime, 3 prime.
Okay, I'm not sure then.
So there is a simple answer. You need not to get confused. To get rid of confusion, we introduce the term prime. So where the confusion is going to be there. So these primes are being used for your sugar. If you remember the structure of your nucleotide, there are 2 rings are there. One ring is present in the sugar, and the second rings are present in your nitrogenous base. So both the rings, both the carbon chain, whatever the type of the carbon is, you have to name them. So you want to distinguish whether you are talking about the sugar carbon or the nitrogenous base carbon. So when we are talking about the sugar, we use the term prime, and when we talk about the your nitrogenous base, we simply use the 1, 2, 3, 4, 5, 6. That's why if you talk about the linkage between sugar and the phosphate, you just simply call the glycosidic linkage. Okay. 5 of nitrogenous base, there you don't use the term prime. So this is the basic simple thing. The usage of the prime is just to distinguish between the sugar and the nitrogenous base. Okay. Okay. So now we continue further. So this is the first requirement. So it must have the 3 prime OH and the 5 prime phosphate group. So it simply means we are talking about the presence of these groups in your sugar. Okay. So in the pentose sugar that you are having in case of DNA, it is going to be deoxyribose. Deoxyribose must have a 3 prime OH and it must have the 5 prime phosphate. Okay. So other than these, some more requirements are going to be there. First requirement is the, as in case of restriction nuclease, some of the requirements are going to be the same or not the same, it's similar. First is the there also you need the divalent ions. So here also you need the divalent ion. And there you need the cofactors. So here also you need the cofactor. So here there are the cofactor. There you are talking about the SAM, but here we don't need the SAM. Here we need the NAD, nicotine adenine dinucleotide. So it utilizes NAD as an energy donor, where the T4 DNA bacteriophage they required ATP as a cofactor. So these are the requirement of it. The role of DNA ligase, as we mentioned, is to seal the nick, or simply to join the ends, or to join the your sugar phosphate backbone. So whatever type, in case of your DNA replication, you utilize it for joining the lagging strands, or simply the Okazaki fragment that you are having there. The joining process is essentially normal in your DNA synthesis, repair also. These all things, whatever we are talking about. In case of restriction enzyme, you mentioned that restriction enzymes were the part of your defense mechanism, part of prokaryotic defense mechanism. But ligases are not associated with the defense mechanism, but they are associated with the replication and the repair. So the ligase enzyme being picked from the your DNA replication machinery or DNA repair machinery. Okay, and the ligation is the only possible is going to have when the 2 strands of DNA they come close to each other. If 2 strands of DNA they are very far apart, then they are not going to ligate. So when they come close to each other, they are going to ligate. So as if you remember in case of restricted endonuclease, we talk about the 2 types of the cut. One is the blunt cut and second is the staggered cut or the sticky cuts. So sticky cut, the fragments out of those are having the overhangs and these overhangs, they can come close to each other and they can pair on the basis of the complementarity. And when the complementarity is there, pairing is going to be there, so then they are very close to each other. So when they are close to each other, then these ligases can join them. These ligases can make the phosphodiester bond in between them. But if they are going to be blunt end, then they are going to stay far apart. So then the formation of the phosphodiester bond is going to be a bit difficult. Okay, so the most widely used ligase is going to be T for bacteriophage. So the enzyme that we are talking about, or the enzyme that we are going to use in our laboratory, is going to be T for bacteriophage DNA ligase, or simply we call it the T4 DNA ligase. What are its requirements? Its requirement is the ATP as a cofactor, and it also whereas the enzyme that you got from the E. coli, that use the NAD as a cofactor. Okay, so what they are going to use them? They are going to utilize them for the energy. Except these, the catalysis mechanism is going to be similar for both the ligase. So only the requirement is going to be different. So requirement depending on what source you are going to get, they may be differ, but the process are going to be more or less similar. What the process is going to be, how they are going to form the phosphodiester bond, we will talk in next slide. Okay, so role of cofactor is splitting and the formation of enzyme AMP complex. So this is the one. So here is the one more thing, the AMP complex is there. So enzyme AMP. So from where the AMP is going to come? AMP is going to come from your DNA strand. So which for the phosphodiester bond between the hydroxyl and the phosphate group, exposing them. Okay, so this is the thing. Mechanism. So how it is going to occur? So this the thing that we have to talk about. So mechanism, as I mentioned, that the first thing is that they must come close to each other. So ATP and NAD. So depending on whatever type of enzyme is you are having, if you are having a T4 DNA ligase, you need ATP. If you have E. coli DNA ligase, ECO DNA ligase, then you need NAD. So what is going to happen? They first react with the ligase enzyme. So in the first step of your ligase, your ATP or NAD. So just I'm calling here the ATP. So you keep in back of the mind that depending on the enzyme, it may be NAD also. So first thing what is going to occur? First, your ATP is going to interact with your ligase. So when it interacts with your ligase, what is going to occur? They form a covalent enzyme AMP complex. So when this enzyme AMP complex forms, this is the adenosine monophosphate. So means two phosphate groups are going to be released from it. Then what is going to occur? Where it is going to link? So it is going to link with the alpha amino group of lysine residue. So this AMP is going to add it over this the epsilon amino group of your lysine. So the lysine residue present in your ligase enzyme, to that this AMP or ADP is going to form a complex, through that we call it the enzyme AMP complex. Okay. So now this enzyme AMP complex is going to now the job is going to activate the 5' end of DNA molecule. So now the job is the activation. So what the activation you have to do? Now you have to activate the 5'. So at the you all know that when we in the DNA replication, when the DNA replication is going to occur, the role of the enzyme, role of the energy is there. You have the ATP forms, adenosine trinucleotides. So DNTPs. So nucleotides are the triphosphate in nature. So you need energy for the formation of the phosphodiester bond. So here this first the job that we are calling the activation, that is going to come from this one. So now the what next thing is going to occur? This AMP moieties, what it do? It activate the phosphate group at the 5' DNA molecule to be joined. So now what it activates, activating. Now it is called as a donor. Now the final step is the nucleophilic attack. So what nucleophilic attack? So all of you know that the what this nucleophile means. What is nucleophile? All of you have done in your first, second, third chemistry you have done. So what is a nucleophile? What is a nucleophile? What is a nucleophile? Nehal, Yukta. Nehal, Vivek. Vivek. Namrita.
Sir,
Yes, Yukta. What is
Sir,
the
am I
nucleophile?
audible?
Yes, you both are audible to me.
electron
Sir, so they are
are
electron donors.
species and they
So speak one by
donate.
one. Speak one by one. Yes, Yukta. Yes, Yukta.
Sir, I said nucleophiles are electron-rich species and they donate a pair of electrons to the electrophile, which are electron-deficient species.
Okay, what do you say, Nehal?
Sir, I was saying that they are the electron donor species that donate the electrons.
Okay, so who are the else were saying about it? Vivek, you are saying something?
Sir, I was just saying that they form these pair. They provide the pair of electrons and lead to a formation of a new covalent bond.
Okay, so all of you are right. So simply as Yukta and Nehal mentioned that they are the electron rich, and nucleophile means simply nucleus loving. So they are the nucleus loving means the proton loving. So they are going to attack the nucleus where the charge is the positive is going to be there. So now the nucleophilic attack is there. So nucleophile is going to be electron rich. So the OH group is the electron rich group. So the 3' hydroxyl group is the electron rich one. So it is going to attack the nucleophilic way, and it attack to the phosphorus atom. So then leads to the formation of your. So phosphorus atom is going to act as an acceptor, and 3' OH is going to act as a donor. And by this process, the formation of phosphodiester bond is going to occur. And the reaction is driven by the hydrolysis of a pyrophosphate. So what the pyrophosphate is there? So what is the pyrophosphate? Can anybody tell me what is the pyrophosphate? So what is the pyrophosphate?
Sir, that contain two phosphorus molecule.
Yes, you are right. That contain the two phosphorus molecule. So as in the first step, we are having this AMP. So we start with the ATP. So ATP is going to have a so simply any nucleotide it is going to have, that is going to have P, and P. So this is the ATP. So the AMP is going to have only one P. So these PP, this PP is called the pyrophosphate. So two phosphates are joined together. So at this step, when this AMP complex is going to form, this pyrophosphate is being released. So this pyrophosphate is going to play as a source of energy in your third step, in the last step that leads to the formation of your phosphodiester bond. So here the diagrammatically we can see how this thing is going to occur. So as we mentioned the first step, so in the first step we mentioned that what is going to occur. In the first step, your enzyme ligase is going to be there. At the ligase at the active site, it is going to have one lysine, and lysine is going to have an extra amine group. So through this amine group, one ATP is going to come, and this ATP is going to form a complex. And to that we call the lysine enzyme AMP complex. So now what this is going to do? This is going to activate the phosphate group. So this AMP and go to join to the phosphate group. Okay, so this goes to the phosphate group. So now this is called the activated enzyme DNA complex. Now what is going to occur at the next step? So the next step, this complex to this, now the 3' OH is there. This 3' OH is going to serve as a nucleophile, and this is going to attack here. So when it attacks here, so this node is going to work on this complex that we mentioned, the enzyme AMP DNA complex is going to broken down, and this node is going to be formed. So here the hydrolysis is going to occur, release of one water molecule is going to be there. So in this process, you need an energy. So that energy is going to be provided by the PP. This is going to be released at this step. Okay, so that what is going to occur. So this phosphodiester bond is going to be formed. Okay. So this step, this thing we call the. So this you know that you start with the G. G is having the 5. This is the 5', and this A is going to have a 3'. So this 5 3' they join. So A is going to join through this phosphate group 3' OH of adenine. So guanine is going to join with the adenine. So simply, so this is the way how your DNA ligase are going to work, and this is the mechanism of action of your DNA ligases. So one more important thing is there about the property of DNA ligase is that the temperature optima. So as we all know that these most of the enzymes are going to have temperature optima of 37 degrees Celsius, but in case of DNA-DFR ligase, it has optimum temperature at 16 degrees Celsius. Okay, so DNA ligase can even work at a very low temperature also. Okay, and even these ligase enzymes are functional at 4 degrees Celsius. Okay, so temperature optimum of DNA ligase mutated ligase is at 16 degrees Celsius. However, ligation is also achieved by incubating at a 4 degrees Celsius by incubating overnight or at room temperature. Okay, so different temperature you can achieve your ligation in different time intervals. So if you want to slow down it, you want to make your ligation in a way that you don't want to hurry up. So like sometimes you are having blunt ends, if you want to join your blunt ends also. So in that case, you can do a process at a room temperature. It can take a bit longer time, but joining should be a proper thing. Okay, so this is the bit some of the things that we were talking about the ligases, and now comes to the job of the ligase. As we mentioned, the ligase means the joining. So the application are going to become wherever you want to join. So the simple application is, as you mentioned, where they are being used, as they are the part of your daily application. So they are going to be utilized there, and they are going to be a part of repair mechanism also. They are going to be utilized there also. But to us, the thing is that how we can utilize them in our recombinant DNA technology. So the simple answer is that we are going to utilize for joining them. So what we are going to join? We are going to join your DNA of interest, your gene of interest into your vector. It's 2 DNA fragments. That recombination that we want to do, this recombination, the combining things we do with your DNA ligases. So in molecular cloning ligase has a routine use, so daily use. We definitely you can say we are having. So without these ligases, your RDT is not going to move further. So this is the second important enzyme, and these two are the most important enzymes. Simply you can say that that you need in your recombinant DNA. Some more applications are there that other than joining them to the vector to the your fragment, you can join utilizing them some adapters, some linkers. Then you want to convert your blunt end to a sticky ends, or simply you may say that the most of these 2-3 are the application of it. So application simply means you want to join. So in any cloning process, any transgenic process, the joining is going to be occur with the use of these ligases only. Okay. So I'm stopping it now at the ligase. So now after this we will move to our next enzyme, So next enzyme is going to be the polymerase. So now any one of you, if you like to have any question on the ligases or the restriction endonucleases, their mechanism, their uses, their function, please let me know. So because today we have some more, enough time to discuss that. We have at least 5-7 minutes to do that. Any query? Anyone have any query?
No, sir.
Nothing, none. So Adish and Manaswini, they have a contract from the class for saying yes or no, and they are dedicatedly doing their job also. Today Manaswini, I think, was not interested in doing that.
No, sir, I've just been having network issues, so I wasn't able to unmute my mic.
Okay. Okay, so ligase is well understood by everyone.
Yes, sir.
Yes, sir.
Okay, just a moment. Yes. कौन बोला था? Who raised the hand? Who raised the hand? Who just joined? Who joined just now? Who among you have joined just now? Okay, no problem. I can check your attendance sheet, so that will tell me that what time you are, because thing is there now the attendance are going to be linked with your joining time. So then in that case you may have some difficulties in that case. Okay, so none of you have any query. So we are moving to our last part, that is your attendance part. So please turn on your camera when I call your name. Just a moment. Okay, Adish. Adish.
Present, sir.
Atoiba.
Present, sir.
Ayushi.
Present, sir.
Demetrius.
Present, sir.
Ishita. Ishita. Janvi. Janvi. Kshitij. Kshitij. Manaswini. Manaswini. Namrita.
Present, sir.
Nandini. Nandini. Nehal.
Present, sir.
Upal,
Present, sir.
Sahiba,
Present, sir.
Sanjana,
Present, sir.
Shakti, Shivani, Tanvi,
Present, sir.
Tanvi,
Present, sir.
Tridev, Tridev,
Present, sir.
Vivek,
Sir,
yes, Janvi,
sir, I'm present due to network.
Yukta,
Present, sir.
Yuvraj, Yuvraj, Nandini, 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 okay, so thank you all. Stay safe and stay happy. So we'll join back next week. Till then, stay safe and stay happy.
Thank you, sir.
Thank you, sir.
Thank you, sir.
And if any one of you have any doubts, still we have 3-4 minutes. We can discuss that. Or we. No doubt, nothing. Everything is clear.
No, sir.
So then we'll see you in our next session.